and you are most welcome to ask any questions about Western blot and antibodies.
Thank you very much. And if you have this workshop is built based upon my experience with Western blot and antibody work, and is built upon questions which I have been receiving over the years because I'm taking care of technical support. If there are any unanswered questions or you have encountered any specific antibody behaviors which you would like to share with me, we can do it in the end in the Q&A session. So as you will see, there will be part 1 covering part of Western blot protocol. Then we will have a 5-minute break with quiz, and you will be able to test your knowledge, and if you succeed, win a free antibody of your choice, which you can choose pick up from our catalog. And then we come back to the lecture, and then we have a Q&A session in the end. If there is any issue that I am not speaking clearly or that I'm gone suddenly, or if you would like me to slow down, please let me know. And you can also type your questions during my presentation in the chat box. This presentation is going to be also recorded, so you will receive the link to the recording afterwards. And let's begin, because Western blot is pretty exciting procedure with many parameters, and I would like to review for you what I have learned during troubleshooting over the last 26 years, which can help you in your work in the future. But first, let's introduce Agrisera. So on a winter day, we have a view like that outside the window, and in a summer day, we have constant light. And we are currently just in that period. We are located 15 minutes flight from Stockholm, so a little bit far north, but still not at the polar circle. The company was established quite a long time ago. We just celebrated our 40 years anniversary. And as you can see here, we have 2 locations, one in this beautiful landscape of northern Sweden, outside city of Umeå, where we have a farm where we are producing antibodies. And in this facility in Umeå, we are doing quality controls, purifications, quality controls, and we are shipping antibodies all around the world. These pictures are taken at our facility, so this is not any Shutterstock pictures. You can see my colleagues on them. In 2020, we became part of Olink, which is a company which built up kits based on antibodies, which allow detection of and quantification of proteins in human blood. Based on few microliter of blood, the profile of various proteins connected to various diseases can be analyzed. And as you see, we have become a part of Thermo Fisher 2 years ago, and the company is ISO certified, which also makes our processes to be very tightly controlled and documented. And antibodies for plants is the focus of a company from over 25 years ago. I am myself a plant biologist. As you heard, I graduated in Poland, in Poznań, and we have been building this plant antibody catalog over this last 25 years based on collaboration with researchers also from Poland and from all around the world. And last year we received this prize we are very proud of. Based on publication record, we have been awarded a supplier which is most successful. We're providing antibodies for plant proteins, hormones, epitope tag antibodies. For those of you who are not working with plants, we have over 200 epitope tag antibodies to various epitope tags, also secondary antibodies, and they have been used and published in over 10,000 publications over the last 25 years. And this workshop is going to be covering, as you see, part 1, we come up to the blotting and protein transfer through blotting, and then in a part 2 after the break, I will cover all other steps, including detection and also result interpretation, because that one can be sometimes quite troublesome. How do we visualize and measure specific protein in a complex mix? Do you know how old Western blot technique is? 10 years, 15 years, 25 years? It has actually been used so long time ago for the first time. This is the first publication where the group is describing this technique, very exciting technique. So we have a very complex protein mix where we are trying to analyze specific protein using antibodies. And why the name Western blot? We have Southern blot, we have Northern blot for RNA, we have Eastern blot for modifications, and then we have a Western blot for proteins. And this is, as you see here, you maybe get a connection to rows of winds from the compass. So we have Western blot because also the lab which first invented this technique was located on the West Bank. And transfer stack, this is the base for the Western blot, and I'm just reminding you, if you're working with Western blot, you have done it, you will know what I'm talking about. For you who are new to this technique, this is just a reminder. So we are having separation of proteins in a gel based on their molecular weight. And afterwards, this is a wet transfer. We are assembling so-called sandwich and apply electrical current, which means that proteins are transferred in a transfer buffer from the gel to the membrane. And then, of course, these windows which are appearing here will contain also links. You will receive this presentation afterwards as a PDF. So I hope you will have use of it in the future, and then these boxes on the slides will lead you to also a little bit more of information. So through this Western blot, what we are aiming is to obtain a copy of what we have had on a gel. This is a principle of the Western blot technique. So we have proteins separated in a gel, and then we need an exact copy by a transfer to a membrane. Remember that exact copy is also a very important part of Western blot technique. I'm sorry with this little bit spooky slide change. I have not been aware that it will make such spooky transitions. I apologize for that. Here is the antibody which is doing the job in Western blot. Just a reminder for you how it's built. So this is like a Y-shaped molecule. And of course, antibodies which we are carrying, IgGs, are looking exactly that way. We have so-called handle, fragment that crystallized. At the time of research on immunoglobulins, that protein was left in a fridge and it precipitated. So that's why it's called FC, fragment that crystallized, and it is unchangeable part of the antibody, which is also used, binds to protein G or A, and it's used to purify total immunoglobulin fraction. The most exciting and important for us is this part. This is where the binding to antigen is occurring. So this is a changeable part on the antibody, which will be adapted to bind to specific region. In our case, we're interested in protein detection, so this part will facilitate the binding to a specific protein. And antigen binding domain, that's the changeable part on the antibody molecule. And what is in that part of the antibody, we have an epitope. And then this is a very important part to understand how antibodies are working in different techniques. So as you see here, we have a linear epitope. Epitope can be from 2 to 15 amino acids, sometimes a little bit longer when it comes to conformational epitopes like this one. But in Western blot, the most important is a linear epitope. So these are subsequent amino acids in protein structure to which antibodies are binding. And these epitopes are recognized by antibodies in Western blot immunohistochemistry, so in techniques where proteins are most likely quite denaturated. And we have also these continuous epitopes. So if you look here, two distant amino acids are brought together by protein fold. And these discontinuous conformational epitopes are recognized in the native structure of the protein and therefore in techniques like immunoprecipitation, also Western blot in native techniques, in native conditions, or CHIP. So please remember, Western blot, it's mainly, we are talking about linear epitope recognition. In techniques where we are working with a native protein, antibody needs to bind to these discontinuous epitopes. And this is a key for successful detection. Just to remind you that we have a clonality also. So we have antibodies which are poly or monoclonal. And there are different misconceptions about which ones are better. It actually depends very much upon in what way they are used. Polyclonal antibody, poly means many. It means that it's a pool of epitope of antibodies which will be binding to different epitopes. So this is a big pool where the antibody pool will recognize fragments, linear fragments, if we talk about linear epitopes, between 2 to 15 amino acids on a target protein. And polyclonal antibodies can be done in different species, as you see here. This is ordered in how much volume we are getting of a specific antibody. So we have from llama to the mouse. And this is poly. Mono means one. So monoclonal antibody would recognize only one epitope. And that epitope can be between 2, 15, 20 amino acids long. Only one. So this can be of advantage in certain techniques, but it can also be a disadvantage. Because if a monoclonal antibody loses reactivity, then it's over. If a polyclonal antibody is losing activity, it still may work, but with decreased efficiency, because it will be some of the antibodies from the pool which will fall off. And monoclonal antibodies are not only made in mice, nowadays they are also made in rabbits, rats, and chickens. Remember, epitope, the binding site, it's few amino acids long. This is where the antibody is binding to the target protein we want to visualize. And antibody choice must match species and application. What does it mean? It means that you need to be certain that the antibody which you are using is actually done to a sequence which is coming from the species under investigation. And this is very often overlooked. Instead of checking if a sequence is found in a protein I want to detect, we run to the lab and spend weeks trying to get Western blot to work. This is where the failure begins, I would say. This aspect is very often overlooked. And it will save you a lot of work if you, before you're using any antibody, you will actually check that it is made to a sequence which is also found in a protein which you are aiming to detect. And the antibody choice must match also application. What do I mean by that? It's actually possible to predict if the antibody is going to work in immunoprecipitation. And I will show you how. So if you do this theoretical analysis before you start experimental work, you will save yourself a lot of time. Please remember about this. This is extremely crucial for your successful detection. So you will save a lot of precious time in the lab if you will remember about this. So how do I choose the right antibody? I will show you an example here. We have a photosynthetic protein, PSBA, and the antibody can be made to a whole protein, but it can also be made to specific peptide. And making antibodies to short sequences makes them more specific. This way we can exclude cross-reaction with other proteins. As you see here, of course, we are not choosing peptide for antibody production in a signal peptide area. We are choosing it in after the cleavage is done. This is a sequence from N-terminal. This sequence is chosen from inside of a protein, and here we have a C-terminal sequence. So before you use this antibody or any other antibody you want to use, you need to be certain that that sequence is found in a protein which you're aiming to detect. It is very easy to obtain false positive results with antibodies, especially when lacking controls. Doing this analysis, you will save yourself a lot of trouble. If it is a commercial antibody and supplier does not provide exact sequence, you need to send your sequence and ask. So please remember this for the future. This is extremely important. Each year I'm finding publications, several publications where Agrisera antibodies have been used on species where the sequence we used for immunization is not even found. So this is extremely scary, I would say. And that sequence the antibody is made to needs to be conserved in at least 70% in linear subsequent order of amino acids to allow antibody to work on your target protein. So how do I choose an antibody for multiple species detection? This is an approach which we are using for last 25 years to save animals. We are designing peptides which are conserved across the wide taxonomic species range. So we are choosing their conserved peptide sequences which will be found in the same target protein in multiple species. Of course, this approach is not always possible, but we are working this way to provide antibodies which are very specific by using short fragments, but also which can be used on a multiple species range. So this is a very neat approach. And if you are using several model species, you can always check on a sequence level if before you go to the lab, if antibody can work on that species or not. Very often it is done opposite way around. First it is tested, and then if it doesn't work, then maybe the questions come. First, you should ask yourself if the sequence which antibody is made to is also found in the protein I am aiming to detect. Here we are showing a so-called quantitative Western blot, where one and the same protein can be quantitated over the wide taxonomic range using quantitative Western blot technique and a specific standard, which is provided together with the antibody. And I often get the questions when the Western blot result doesn't look ideal, like these bands here. Will knowing antigen sequence help to identify cross-reacting bands? And the answer is no, because each of such bands contains hundreds of other proteins. And sequence will not tell you much. Only mass spec analysis will tell if which protein can be identified. You can learn here. So knowing the sequence, you may only predict if the antibody is going to work in your species, but you cannot predict any other bands which are showing up on Western blot. And before any antibody is produced, especially to plant protein, we are conducting so-called pre-immune serum screening. And I've been asked for that. So here you will see we have samples from barley, Arabidopsis and barley, which were run, separated on a gel and transferred to a membrane And then we have serum from non-immunized animals. And as you guess, animals are eating plant-based diet. So the protein which will be showing up at 50 kilodalton is Rubisco. And this is a problem. If such an animal with a background already to Rubisco would be immunized with a protein of with a peptide found taken from a sequence of a protein which is around 60 kilodaltons, the antibody is not going to be useful because the cross-reacting signal from Rubisco will be already there. So when we are choosing animals for immunization, we will be looking at molecular weight of any cross-reacting bands and choosing the animal for immunization with immunogen, which is at least 20 or 30 kilodalton away from any cross-reaction. If you look here, in that animal, it was not immunized, but it already has antibodies to some target in a plant extract. So this is a very important step to secure the quality of the obtained antibody. And yeah, there was a poster about antibody production. If you're curious, it is available on our website. And then how the antibodies are working in Western blot. We have one option when a primary antibody is labeled with an enzyme or fluorophore. So antibody is binding to a protein which is transferred to a membrane and the signal is recorded. But we have a second option when, I was too quick, sorry. So there was a first option, and then now we are having a second option when we have a secondary antibody coming up. So we have primary, and as you see here, it's not one-to-one. It's more molecules of a secondary antibody binding to a primary, which means that the signal is going to be enhanced. It is especially important for proteins of low abundance, and I will talk about it more a little bit later. So this is the Western blot workflow, which you know has many steps. A lot of things can go wrong. And here on this slide, you can see how it actually works in practice. One and the same antibody has been sent to 4 different labs, okay? Each lab is using Arabidopsis for detection and the same primary antibody, and chemiluminescence, as we can see, as detection method. So first lab says it doesn't work, no signal at all. Other labs get a lot of background, and the lab number 4 is providing us with a result. So each of them was using same antibody and theoretically the same material, but we are working here with detection of a transcription factor, which is a little bit more tricky. And that laboratory was using nuclear fraction, which is necessary to visualize, to concentrate this protein and visualize it. How much do you know about the protein you aim to detect? That's extremely important when we are starting with the Western blot procedure. I would suggest that you don't even go to the lab before you analyze this. So here is your checklist. Biochemical properties, isoelectric point, solubility. If you extract a protein at pH of a buffer close to isoelectric point of your protein, it's not going to be soluble, right? Expression level is very important. It's more difficult to detect proteins of low expression. Organ tissue localization, crucial thing for proteins which are low abundancy, and cellular localization. So you need to start with this checklist, not with your experiments. You first need to learn biochemical properties of your protein which you are aiming to detect. Amount of target protein in a tissue can actually be checked in a database. We have been learning over the years that when proteins are present in less than 10 parts per million in the tissue, the detection is going to be extremely difficult. So we actually are not producing commercial catalog antibodies to proteins of so low amount in the tissue, because detection is going to be very difficult. And in this wonderful database, which is called Protein Abundance Database, you can check for different species, for human, for rat, for Drosophila, for Arabidopsis. You can check how much, based on experimental data, how much of a given target protein is in the tissue. This is extremely useful database because you can also find a material which you should use, which has increased level of your target protein. Sample quality is of extreme importance when you are working with Western blot. So please, please don't use old samples. If you use samples which are more than a couple of months old, there is a big risk that your target protein is going to be degraded. And we need to be having samples of extreme good and high quality. Here it is if you're working with plants. We are having on our website images of model species and pathways, which you can download. We are focused on plants, therefore the plants. And high quality requirement of protein samples can be achieved in different ways, but you need to work fast. You need to, for proteins of low abundance and sensitive proteins, the best is to analyze them directly on Western blots after extraction, not to store them. And time-efficient extraction method is a bead beater. And of course, the protocol needs to be optimized depending upon which material you're working with. Is it E. coli or is it diatoms, which have much more robust cell wall? We also have other methods which are a little bit more time-consuming. Sonication is a heat-generating procedure, and mortar and pestle. These methods are not high-throughput extraction methods. Of course, it's possible to extract 20 samples using this method, mortar and pestle, but your hand will get tired, and the efficiency of extraction will not be the same comparing sample 1 with sample 20. So please keep this in mind and choose the most appropriate method for your extraction. And you need to work really fast and very precise. And on a conference, we have sent some protein extraction posters. You can pick it up if you haven't done it yet. It should be displayed. If there will be not enough posters, you can always order them from our website or download them from our website. If we work with proteins of low abundance, fractionation is a must. Please look here. Very many antibodies have been discarded when they were analyzed on a total cell extract, because for proteins of low expression, there will be no band. Here we are showing how fractionation concentrates the target protein. This protein was chloroplastic. So in photosystem 1 fraction, we obtain the best signal. So if you work with proteins below 10 ppm, you will not run away from need of fractionation. It's a laborious procedure and takes time and skill, but you can know it in advance before you even go to the lab. And protein extraction methods involve many different buffers. And here I want to show you, it was one of the test labs of Agrisera antibodies. They used at first trees-based extraction buffer, and they modified it, included hippies, and then target protein band appeared. Extraction buffer can be of extreme importance in some cases, and sometimes you need to include urea to unfold the protein and make it accessible for the antibody. On this blot, we have so-called garden blot because we have samples from barley, Arabidopsis, and rice. Sequence is perfectly conserved in all 3 species, but the antibody is not detecting target protein unless the highly denaturing reagent is included. So sometimes such modifications can make a difference. So here is the checklist for you for the future, how to work when proteins are of low expression, or if you don't have a signal, then you should go to PAX database and check protein abundance first. So as you see, quite a lot can be done before you even go to the lab. And good laboratory practice, GLP, is a must when you're working with a Western blot. So you keep it clean. You need to clean the equipment very well. We always work in a coal and we are precise. So when your pipettes were calibrated last time, this should be done at least once a year. And gel electrophoresis, we start with the first step. What do we load per well? What are you loading? Are you loading protein or are you loading volume? This is first key step for your success, because this is what we are using. We are using Novex gel system from Thermo Fisher for the last 25 years, which works very efficiently. And we are loading microgram of protein or chlorophyll for photosynthetic organisms. I know that many labs are loading on volume, and this is wrong, because you don't really know, especially for proteins of low expression, you don't really know how much in micrograms of protein are loaded on a gel. So I often hear that, but it's not possible to estimate protein concentration in the extraction buffer or loading buffer, and that's actually wrong, because there are reagents which will make it possible. So there are reagents which will allow you to estimate protein concentration in presence of for example, the naturons or urea. So please start loading based on protein or on chlorophyll, because your extraction is not perfect. So you cannot assume that when you extracted 10 samples, it will be always the same protein concentration in them. And also your material maybe comes from different physiological conditions. So please remember, always use a determination of a protein in your sample. And I received a question if it should be loading nanograms per well on micrograms. It depends what you're loading. If you're loading recombinant protein, then it should be nanograms. If it is total cell extract, it should be micrograms. And here you can see fine-tuning of the Western blot when researcher was adjusting protein load versus antibody dilution. So you can see here that by playing with protein load per well and primary antibody dilution, you can actually make background bands to fade away. Because Western blot is about signal to noise ratio. It is about how fast the signal comes for the target protein compared to everything else. And also we can use 1D or 2D gels for separation of proteins, and it will be a difference between them. And there was a question how this affects the results. Of course, it depends what questions you are asking. Most of antibodies in Western blot are used on a one dimension. So we are loading protein at top of a gel, applying electrical current, and then we separate them based on molecular weight. So each of those positions contains hundreds of different proteins. Using second dimension, we can separate each of these bands into specific spots. So Western blots can also be done on these type of gels. But of course, there is a difference between 1D and 2D separation. This is for your future information. And it depends which gel you're going to use. It depends what questions you're asking, and that determines the choice. And sample denaturation can be done in different ways. And probably some of you is using 100 degrees, 90 degrees, 70, 35. As you see, we are preferring 70 degrees for 5 minutes. And the reason for it is that some proteins aggregate when heated in high temperatures, and they will create aggregates which never enter the gel. Therefore, lack of band with antibody, lack of antibody detection. So I would encourage you to 70 degrees for 5 minutes, which is a good denaturation method, which does not create aggregates of proteins. And now we're diving into transfer. Here you can see a wet transfer. Please remember that it should always be done in a cold. This is how we do it in our lab. It can be done either in ice bucket with cold buffers or in a cold room, but always in the cold. And protein transfer conditions depend upon size of a protein. Have you been thinking about it? It's depending upon molecular weight of your protein, you need to adjust your transfer conditions. So here you see the difference. Each lab should actually have both membranes of different pore size, because for larger proteins, we are using larger pore size. Also, larger proteins love SDS, in contrary to proteins of lower molecular weight. And wet transfer is recommended for proteins of high molecular weight. So as you see here, this is another parameter which you should check before you go to the lab and do the Western blot. There's no one protocol fits them all here. Which membrane to use? I can bet that most of you worked with PVDF membrane, because that one is the most popular membrane. However, there are differences between those two membranes in binding properties for proteins. Both can be air-dried and blocked and used later. Nitrocellulose membrane is a little bit more fragile compared to PVDF. Some protein antibody pairs will give better results on nitrocellulose compared to PVDF. So it is not possible to say which of them is better over the other. So both membranes are available in different pore size. Please keep this in mind, because this can also be helpful to optimize your results in the future. And membrane activation can be done in different ways. Methanol, which is more toxic, can be replaced by ethanol, especially if you're pregnant and working in a lab. That can be important to change to less toxic compound. And one can do a simple comparison if results are consistent. But there is a possibility to change to less toxic ethanol. Protein transfer type. There are two transfer types, as you know. So wet or semi-dry. And they differ in those parameters. So they differ in amount of buffer, electrode contact, and speed. And wet transfer is a little bit better for beginners. Semi-dry transfer require more skill. So it's wet transfer is less prone to error. So if you're a beginner, I would suggest to work with wet transfer. If you don't have this possibility in the lab, ask someone to teach you, to show you how to do a semi-dry transfer. Protein transfer is crucial later for our protein visualization. And this picture shows you how we are not doing to remove air bubbles, because why should we bother even about air bubbles? If the air bubble is in the membrane, between the membrane and gel, in the molecular weight range of your target protein, there will be no transfer to the membrane in that area. So we are not removing air bubbles this way. We do it inside the cassette. And also, if there are air bubbles on the top and our target protein is in the bottom of a membrane, we do not bother to remove them. And here's even linked to a short video how we are assembling a sandwich, because if you do it for a wet transfer in a cassette without moving around too much between rubbing the gel towards the membrane, the bands will have better sharpness. And protein transfer, quality of protein transfer can be checked after the transfer. And this is done by a simple and quick stain, and I advise you to do that, Ponceau or Kumasi. And of course, if we see that our membrane looks like that, we do not proceed further, because there was a clear issue with the sample. So proceeding with such a membrane with other steps of the protocol will be actual waste of time. So doing this transfer check on a membrane, you can assess that quality of a transfer. So markers are important, yes, but you should also see how is the situation in the other wells of on the membrane. And the other important thing to check transfer quality is to stain the gel. It is also very quickly done. So you basically take the gel after the transfer and stain it in Kumasi. And then why do we do it? We do it to see if all protein was actually transferred from a gel to a membrane. As you remember on one of the first slides, what we are after is exact copy of proteins separated in a gel, which should be transferred to a membrane. And here it's an example of double sandwich. And as you see, we stained the second gel, and it actually had proteins. So the transfer was not accurate for the second gel in the sandwich. So if you have proteins still in a gel after the transfer, it means that transfer conditions were not optimized, which may mean that you will have too little protein later on the membrane for antibody to bind. This is extremely quickly done, and I encourage you to include it in your procedure. Primary antibody format can be serum or purified. So antibodies can be purified to total immunoglobulin fraction or to specific antibodies. Here you can see lyophilized antibodies in the tube. That format is making it safe in case antibodies are transported and await on customs for a week or two. They will be protected and not degraded when they are lyophilized. And here on this slide, you will see how much of the specific antibodies in the serum. We often get this question. It actually cannot be determined until it is purified and we can calculate it. So please be aware, some suppliers are selling, or also Agrisera has some antibodies which are provided in total immunoglobulin format. And then you need to keep in mind that it's written 1 milligram on a vial, for example, but only 0.5 to 5% of this 1 milligram is going to be your specific antibody, because we are dealing with total immunoglobulin fraction. If we purify the antibodies on specific columns, then we have them immunogen, antigen purified, and then we will have most of the antibody in the tube is going to be specific antibody. And I just want to remind you how the purification is done and show you how we do it in our labs. So we are having columns through which the specific protein or peptide coupled to the column which antibody was made to, and then we are passing through the serum and purifying specific antibody. So it looks like this. First, we couple protein or peptide to the column. We apply serum. Specific antibodies are binding. The rest passes through. And then elution is usually done with low pH, and specific antibody fraction is collected. So such preparations are 95% pure. These antibodies are expensive, and many times we want to reuse them. And you probably maybe also done it, but you reused primary antibody. And I often get those questions. So how many times can a given antibody be reused? Some antibodies only once, some 20 times. It is very specifically primary antibody dependent. So simply, if you have not gotten the signal after reuse, it means that whole antibody bound in the first incubation. there's no more to bind, and therefore such antibody cannot be reused. And please do not reuse secondary antibodies. Two primary antibodies, sudden, if they stop working, this can happen. Antibodies are biological molecules. This part here is the variable part, which is influencing stability of a whole molecule. Antibody stability cannot be predicted in advance. We have antibodies which are stable now for 40 years of storage, but there are also such which lost activity after 1 or 5 years. Other factors which include is of course storage temperature. If a freezer broke, there can be also deterioration in antibody reactivity. Also, sometimes you need to add protective glycerol or BSA. If you buy catalog antibody, it should be specified on product information sheet. So unfortunately, antibody stability over time is not possible to predict in advance. And now comes the break, and this is a QR code to the quiz. So I encourage you to test your knowledge, and we will meet again, and will be second part of my lecture in 5 minutes. So we say we meet at 12:20. I can also paste the QR code on the chat, but I hope that you would like to participate in this. It's just, I think, 9 questions, and the prize is a free antibody from our collection. So we start again in 5 minutes. Right. I hope you enjoy the quiz. I see quite many of you completed it, and you are welcome to do it 2 or 3 times. If you would like to be having a chance to win a free antibody, it's completely fine. Let's proceed. So, what are the next steps in the Western blot? We have stopped on a transfer and checking quality of a transfer. And here is the membrane blocking, which is also a often overlooked procedure. So we are not paying attention to blocking. And here a tip, don't use the tweezers as we did here without flat ends, because it can damage the membrane. And of course, there are different options of doing the blocking. It can be 4 degrees overnight or room temperature. As you see on the next slide, it is quite antibody dependent. But I would also like to stop here and show you a method of actually storing the membrane for the future. So we are preparing membranes in advance and drying them protein side up and storing them in plastic bags for 6 months and longer. So it's actually a way to store your precious samples. It will be less risk that they will degrade when they are stored in this way. So please keep this in mind for the future. This is a very good procedure. Once we made all the steps and blocking, but the antibody we wanted to use haven't arrived in time. So then we also dried that membrane and then used it 1 week later. Of course, if you're using PVDF, you need to remember about activation of the membrane. So here is the procedure. It's described how you can do. So you will have it for the future in your PVDF presentation. And now the blocking. So what happens there? Blocking reagents mostly used are 5 to 10% non-fat milk, and then we have commercial blockers, which of course have proprietary content. If you need a non-protein blocker, you can use this PVP40, which is used in fractionation also, BSA, and gelatin. As you may guess, this is a list of from what I have seen over the years, the quite messy and least efficient was gelatin. However, you need to keep in mind that some protein antibody pairs may actually work better with certain blockers. So nothing is written in stone. It cannot use one and the same protocol with antibodies always, because it always depends upon your target protein and antibody. And here, look at this example. The lab returned the first result, this one, and said, non-specific antibody, it doesn't work. I asked them to increase the blocker and blocking time. And look, the background went away. So what was it a problem? Was it a problem with primary antibody, or was it a problem with the protocol? So in this case, increasing the blocker concentration and time of the blocking solved the problem. Sometimes we need to block overnight. So please remember, it is dependent upon specific protein antibody pair. And also, you need to remember that milk cannot be used if you're using this avidin, streptavidin detection, because there will be an interference. And sometimes we have the background, unwanted background, and there are other bands coming, and we cannot get rid of them. And here is a method which is also described on our blog, where you need to have a sample from a deletion mutant. So you need to have a sample where your target protein is not present. And you prepare membrane strips with this sample and then incubate it with your messy antibody. The principle of this is that the cross-reacting antibodies will bind to those cross-reactions, but as there is no target, your target will stay in this incubation buffer. Your antibody to your target will stay. So you absorbed unwanted antibodies. Sometimes people who are doing reusing antibodies to plant proteins, they tell me, oh, with each incubation step, Rubisco background signal is weaker. And that's what is exactly happening. It will be depleted by each incubation. And here you simply need to have a sample from mutant, from knockout, that doesn't have your target to be able to use this method. It is described here, but this is for your future reference. So I'm just moving on here. If you won't have any questions about this procedure, you're always welcome to contact me. Membrane blocking and background after stripping. That was also a question which I received, that why is it stronger background after stripping? This procedure is not my favorite because you have a target protein on a membrane and then a primary antibody binding to it, and then you want to use that membrane with next antibody. What is done by stripping using harsh chemicals, you may actually strip not only the primary antibody, but also the target. So this way, you will change signal to noise ratio, and therefore higher background will be observed when using the membrane again after stripping. But of course, it will depend upon how much target there is. Remember, target is always the crucial thing. If it's little of it on a membrane, it will be much more difficult to visualize such a protein. And comparing antibodies, does it work? This happens very often that, for example, actin, which is a very abundant protein, an anti-actin antibody is compared to an antibody to transcription factor. This is wrong. You cannot do that because there is a difference in amounts of these proteins, so that the signal for actin will be visualized much faster than for the other antibody. And each antibody is binding to different part on the protein and is unique. So I would advise you against any such type of comparisons. Here is for your future reference, more information you can discover later. Antibody incubation. There are a few methods if you want to save time. So in this one, as you can see, 3 primary antibodies were used at the same time. Researcher had little sample, so instead of doing strippings and used 3 different primary antibodies in the same incubation. It works, but you need to work with antibodies you know, how they behave. And also, as you see here, it needs to be at least 20-30 kilodaltons between those proteins. So this is one of the methods to save time. It's a parallel incubation. The next method is incubating everything at the same time. It may look very crazy to you, but it is actually possible for antibodies to proteins of high expression, which is a lot of in the tissue. As you can see here, we incubated primary antibody, secondary and blocker, everything at the same time. So think how many hours you're going to save from your protocol. So already after 30 minutes, 15 minutes, 30 minutes, we had a signal. But this is not a method for transcription factor. This is a method when you want to shorten your protocol to be used with antibodies to highly expressed proteins. And then we have a third method when we can use directly conjugated primary antibody. There are catalog antibodies which can be purchased directly conjugated, and that way you will save the time on the incubation with secondary antibodies and washing steps. So there are actually some tricks to speed up your procedure. And when it comes to secondary antibodies, please never incubate them overnight. And the incubation time of a secondary antibody is enough if it's 30 minutes to 1 hour. And secondary antibodies are used depending upon which host the primary antibody is coming from. And here you will see the difference. So the secondary antibody is this principle that we have primary target protein, primary, and then secondary with an enzyme attached here in case of chemiluminescence, for example, or fluorophore. And there can be a difference in how secondary antibodies work. Because as you see here, the intensity of your target, the detection, will be much stronger if you have a highly efficient secondary antibodies. And usually we pay attention to how much it costs. 1 milligram costs 200 euro. You should instead pay attention to dilution the secondary antibody can be used in, because that will save you money. And sometimes a secondary antibody may last whole your thesis. For example, Agrisera goat anti-rabbit can be used 1 to 25,000. So 1 vial, 5 years of Western blots, guaranteed. So please remember, don't concentrate on price. Concentrate yourself on price so much as on the dilution in which you can use the antibody. And here, a little bit of a question for you. So we have those 2 secondary antibodies, and which one would you use on an E. coli sample or on plant sample? The difference between them is that this reagent is highly absorbed, and this secondary antibody is passed through a columns to yield minimum cross-reactivity to bovine and rat IgGs, human goat IgGs. So for a plant or E. coli extract, which of those 2 would you choose? Okay, now comes the answer. The correct antibody which would be used would be, that one. So the simple one. Only if you work with human samples using adsorbed secondary antibody to human proteins or human IgG would secure less background. This is why some secondary antibodies are offered adsorbed. This is also important in immunolocalization studies. And now we are coming to visualization. So we have chemiluminescence, fluorescence, or chromogenic detection. And there are some differences between them, and I will shortly walk you through them. All 3 can, of course, be used to visualize our target protein. Here, it is, of course, chemiluminescence, which is shown. There are different advantages between them. So here we have a chemiluminescent detection, but something went wrong in this blot, right? You can see it, I hope, because we often want to make things too good. And we are dealing here with highly expressed photosynthetic protein. And in this blot, it was too sensitive, the ECL, which was applied. So extreme low femtogram detection range is not necessary for protein of moderate and high expression. So when you're choosing your chemiluminescent reagent, you should also consider that. Definitely, chemiluminescence is still the most popular detection method, and it is suitable for quantification. Disadvantage is the fading of the signal. And sometimes there will be background. You may be seen such blot with chemiluminescence. And here you have a checklist what can be a reason. If you are wrapping the membrane in a plastic, that one can lead to increased background. Usually it is that you may have too little of your target, because remember, Western blot is about signal to noise ratio. If you have too little of your target, everything else will light up. So what I would do when having such a result, I wouldn't run to the lab and do next blot. I would actually sit down at the desk and analyze my protocol, finding ways to improve it. Sometimes with chemiluminescence, we can see those ghost bands. So what is that? You may be seen it. It's too much of everything, and reaction goes too fast. And therefore we have a white band instead of black. So to solve this issue is to use less of everything, less of primary antibody, less of secondary, and maybe less sensitive detection region too. And I received this question: if something went wrong in the last step of my detection with chemiluminescence and I didn't manage to record the blot, I record the signal on the blot, can I redo it? What should I do? You can just wash the membrane and do detection, apply chemiluminescent reagent again. And that way you can actually save your experiment within the same day. And now you come to fluorescent detection, which has advantages. As you see here, we have two different primary antibodies, and they will be giving us different signals, different colors of the detection. This is multiplexing. This is a very neat technique. And the advantage of fluorescent detection is the signal is very stable. It's not like in chemiluminescence that it will fade away. So this one is suitable, fluorescence is suitable for quantification and multiplexing. And then we have a third method, it's colorimetric detection, where detection is, we don't need a CCD camera, it's just recorded by eye, and it is very cost efficient. We can put the blot in our lab book, and unfortunately it will fade over a few years, the bands will fade, so it's always good to make a picture. However, disadvantage is that this method is not appropriate for proteins of low expression. The lack of a band may be that you are using not appropriate detection method. And when it comes to the summary, so we have those three detection methods. Chemiluminescence and fluorescence are having high sensitivity. Oftentimes, phosphomodifications, people tend to like to use chemiluminescence as a very sensitive method. Colorimetric method works well, is cost efficient, but please don't use it for transcription factors because it's not going to work. How to get an intense signal from Western blot using a weak antibody? That was one of the questions which I have received. So instead of running to the lab and doing next 10 Western blots, what do? You sit down and you ask yourself, according to this checklist, do I have correct sample type? Do I need to fractionate? Do I have enough protein loaded per well? Is transfer optimal? Primary antibodies, are they purified? Detection regions, are they appropriate? So all this is called optimization. And it's not possible to overlook it, I hope now, after you heard my lecture. So here you can see the difference in optimization, what a single change of one parameter can do. You see here, oh, bad antibody, a lot of background. Okay, what happens when we optimize? The only thing which was changed here was protein load per well. Easy and quickly done. And result interpretation, I squeezed into my presentation because I think it's extremely important that I flag for you certain important point. Of course, everybody would like to have results like this, which can be included in a publication. And I hope with the help of this Western blot workshop, your results will improve in the future. The crucial is when we are interpreting the results to know what is molecular weight of a target protein. We are using markers, colored markers, mixed with markers which will allow us to be visualized during detection, so-called magic marks, for example. And these are having IgG binding sites and very nice span in molecular weight, and they will be visualized during your Western blot development. They can also be used for chromogenic detection. So this allows us to secure that we know where our target protein is. You may think I'm talking about trivial things, but I've received results which were misinterpreted because the markers were wrongly marked. So these markers are not expensive, can be used more diluted than on a supplier's page is specified, and they will help you to secure that you know exactly the position of your target protein. Sometimes calculated and apparent molecular weight of a target protein is not the same. In this example, we have another photosynthetic protein. As you see here, calculated molecular weight 80, but apparent molecular weight 55-60. Why? Because the protein is very hydrophobic. So migration of a protein on a gel can be influenced by these modification, post-translational modification, amino acid composition may change migration by 10 kilodalton. Sometimes aggregation, epitope tags, of course, will also change mobility as well as SDS page conditions. And here is a checklist for you, how post-translational modifications can influence the molecular weight, which is visualized on your blot. So this is extremely important. Besides, we shouldn't rely only on molecular weight when we are interpreting the result and working with antibody which has not been validated by a supplier. Of course, we want to include also loading and normalization controls. And here you have an example. But takeaway from this slide is that you need to know and characterize these antibodies before, because as you see here, they are used in different dilutions. And also, loading controls cannot just be chosen from the catalog. They have to be actually, protein load per well needs to be equal, of course. Bands cannot be distorted. Linear detection range, so not too sensitive ECL, for example, shouldn't be used. And also, this is the crucial point. You cannot just pick up a housekeeping protein because it is supplier offers it in a catalog. This does not work like that because we have different loading controls. You will recognize the names here: tubulin, actin, histone. They are universal for plants and animals. However, they cannot be just chosen based on it that others published them. I want to be very clear here, and I will show you the example. So you see here, actin and tubulin are not always optimal as loading controls. In these experiments, metal stress, and what happens? The actin level doesn't stay the same. So actually mRNA profiling has to be done before we are choosing the loading control. And here is a checklist for the future of other loading controls, which can be, of other internal controls which can be applied. So the ideal Western blot result, we have a band of correct molecular weight, which is gone in the deletion mutant and present in the overexpressor. And this ideal result, provided actually by a laboratory from Warsaw, is actually not happening so often. I'm sorry to break it to you. This is optimal result, but unfortunately, many antibodies will not pass this validation. And even if you're buying an antibody from a supplier, but you have appropriate controls, it is very important to include them. Because as you probably know, the level of validation is different depending upon the company. So here I want to show you a beautiful Western blot result, which was misleading. Look, target protein is here. It's not that one. So it's important to be very vigilant when you are analyzing your Western blot results. And here it is often thought that if antibody is detecting recombinant protein, like this one, a very good detection of a recombinant protein is going to be also suitable for detection of endogenous protein. And look here on this example. Of course, recombinant protein is much more off, so it will be detected with higher intensity. It's uncomparable. And sometimes antibodies will detect recombinant protein, but not endogenous in the tissue, and opposite way around. So if a supplier is providing you data of recombinant protein detection, it does not mean that the antibody is suitable for endogenous sample. Please be very observant, because many providers are only showing these data, and then you assume that such a detection is to be expected in your sample. This is not the case. And I need to move further so we finish on time. Here is another example, the opposite way around. So we have here the antibody is detecting beautifully the native protein, but here we have that protein overexpressed. And as you see here, It's just non-specific background in plants which are expressing this PIP2 protein. So that antibody detects native protein doesn't mean that it will detect a recombinant one. In some cases, the band in the mutant is not gone. And it can be different reasons for that. So one should always check such transgenic organisms because it can be different reasons for it, of target binding, incomplete mutation, gene compensation, and so on. So, but it actually happens. A part of antibodies which we validate, which are validated for us in different laboratories, will actually yield also a band in a mutant. But remember, it can be a band of coming from different reasons. So it doesn't always mean that the antibody is bad, but such antibody, of course, will not be so useful. And when the reasons are for this detection that another protein with related epitope will also be binding an antibody. And you could apply this depletion protocol here, but of course it means also more work. But if we don't have a mutants, what can we do? We can do fractionation. Look at this beautiful example. So this is a photosynthetic protein, which is crucial. It's not possible to get the mutant. So we instead are validating using fractions. So this protein is not present in the stroma. It means that this is a negative control. So even if we don't have the mutants, we can still find another way, another control to validate the antibody. And IP combined with MS is, of course, the most strong validation point for the antibodies. How to be sure that an antibody is binding to correct target protein? Apparent molecular weight. Is it correct? Western blot protocol needs to be optimized. Positive and negative controls need to be included. Linear detection range. Don't use too sensitive detection reagents. Other approaches also included. And obtained results needs to be reproducible. So this is your checklist for the future. And if you are experiencing these issues, poor results, wrong molecular weight of a target protein, lack of reproducibility, worse result with each blot, and you're really tired, then I suggest that you do this. This is a template for consistent reporting of your Western blot results. I'm not troubleshooting antibodies if I don't get a full description of experimental conditions. As I hope you understand, now after this lecture, each of these parameters influence the outcome. So how much of a protein? Was it extracted freshly? Was specific species organ included? Buffer? Then also blotting. Each of these parameters are of crucial importance. So sometimes there are coming students sending their blots and they have been spending a lot of time. And instead, if one analyzed the parameters before starting to change them, one could get a solution to this problem much faster. Antibody validation strategies. These are checklists for you for the future, because there are different approaches which can be applied. As I showed you, if there are no mutants, there are always solutions how to validate the antibody. And if you are buying catalog antibodies, please be careful and check first how those antibodies are validated. And if something doesn't work, contact producer with questions. Last point for the future for you is that how you're reporting the antibodies in your publications is very crucial for reproducibility. Unfortunately, there is no worldwide standard, which means that more than a half of antibodies used in research cannot be properly identified. And this is dreadful because many suppliers have several antibodies to the same protein. And if product number is not reported, it will be very difficult to know which exactly antibody was used. So please take this with you for the future. And if you're curious, we are having this technical blog with a lot of information. It's based on troubleshooting, which I'm getting every week, troubleshooting antibodies which are in testing, or Agrisera catalog antibodies. You signed up for Agrisera newsletter, you're going to get informed about these technical blog posts. And if you're a plant person, we have these wonderful resources, educational posters, images of plants, and now also stickers with different plants. And if you're a plant scientist, please take a look in this global plant events calendar, which Agrisera is running together with American Society of Plant Biology. And of course, reminder of a poster, Western blot poster, which you can grab if there's any copy left. If it isn't, come to our website and order it. And at the end, a surprise for you, we have 100 free antibodies to different targets. You can check under here if any are suitable for your research. There are also epitope tag antibodies. I'm searching for a lab which would like to use antibody to MIC tag in return for results. So if you are interested, please let me know. And if you want to contact us, here are the correct email addresses. My email is very easy, joanna@agrisera.com. You are always welcome to contact me because I am very keen to help. And with the picture of surrounding of Agrisera right now, we have a very intensive summer, soon celebrating middle of the summer in the 21st of June. I would like to thank you for your attention, and I welcome any questions.
So first of all, let's thank our instructor today, so Dr. Joanna Porankiewicz-Asplund, please. Applause, please. And do you have any questions?
If you are shy, you can type them in the chat.
Oh, first of all, we would like to thank you so much for such a beautiful presentation and information about Western blot. It was really fascinating, and we for sure would use this knowledge in our future lab work. Can you hear us?
Yes, sure. Yeah, I'm just writing my email address. So if you would like to eat lunch now or don't have questions right away, you can send them later. You will also receive next week a link to recording and the PDF of the slides with different links. And I hope that this material will help you not to struggle with the Western blot, but to have fun with this technique and get very useful results for the future. So thank you for your attention, and let's keep in touch about Western blot. You are always welcome to contact me.
Yeah, we thank you so much, and applause for the last time.
Thank you very much, and have a good conference.
You too.
Thank you. Bye.